in case of the gene regulation, the third paragraph NCERT, the gene regulation in case of the prokaryote, you see the control of rate of transcriptional initiation itself a predominant target site for control of gene expression. The transcription initiation itself a site for control of the translation, because transcription translation almost simultaneous. Because in prokaryote, the transcription translation occurs in a single compartment. There is no demarcation of nucleoplasm and cytoplasm because of the lack of membrane. Why transcription translation almost coupled? Because translation begin much before mRNA fully transcribed. Why? Any processing is not required for mRNA in case of prokaryote. Why? Because the genes which are non-split. Next. In case of the prokaryotic DNA, that is in transcriptional site, it is a given transcriptional site. It is in this accessibility of it is RNA polymerase. The RNA polymerase is going to polymerize ribonucleotides. The accessibility of RNA polymerase at a given the promoter site, it is regulated by it is accessory protein. This accessory protein works positively, otherwise it is negatively. That means it allows the RNA polymerase to bind at the promoter, or it do not allow the RNA polymerase to bind at what is the promoter. So accessibility of RNA polymerase in a transcription unit at a promoter site regulated by accessory protein. So positively, what do you call it is activators that allows the RNA polymerase to bind at a promoter to proceed for transcription of the structural gene. And negatively, it do not allow RNA polymerase to bind at a promoter. As it do not bind at the promoter, it do not detect this start site. It is the start site. It is the start site. So, regulatory protein affect RNA polymerase in detecting start site. The start site is nothing but it is structural genes. Okay, next. In prokaryotic DNA, the accessibility of the promoter for RNA polymerase regulated by this regulatory protein. This regulatory protein, it binds at operator. Once this regulatory protein binds at operator, it blocks the way. So RNA polymerase do not bind at promoter, do not proceed to carry out the transcription of the structural genes. So in most of the operons, the operon is a group of genes operating together for a common function. Each operon having its own repressor as well as it is operator. In case of the lac operon, it is having lac repressor and lac operator. So repressor protein is produced always in an active state. In its active state, it binds at the operator. So there is no accessibility of RNA polymerase at the promoter site. So it do not involve in the transcription of structural genes. And it works positively. That means the regulatory protein, it allows RNA polymerase. This is the RNA polymerase. It binds at what it is the promoter. So it detects what it is the structural genes that start site and proceed for the transcription. What is formed? mRNA is formed. And as the transcription, translation which are coupled and simultaneous, ultimately it is the protein is formed. So regulatory protein affect RNA polymerase in detecting start site. It acts positively or otherwise it is negatively.